With that, I'd like to welcome you to the Meet Up and Do Science What's the Buzz? Today is Thursday, July 9th, and we're starting just a moment after the hour. And my name's Cassie Sofing, and I'll be pushing through the slides. Just a sneak peek about the webinar that will be coming up next month. In August, the Mission Mosquito team has been working with the Bzzz students out of the University of Texas at Austin, and they're doing a summer internship called the Bzzz Mosquito Mappers high school interns, and we have several projects that they have been working on over the last 8 weeks, and taking the opportunity next month to share with you their research interests, which vary all over from Python programming to mosquitoes to land cover to mapping. So that will definitely be an interesting webinar to attend.
Well, thank you so much. I'm really looking forward to that. Wanted to give you a little bit of information about how many observations all of you and millions of other people from all over the world have been sending in to us. So when you take a look here, from the time we got started with the mosquito habitat mapper to the present time, you can see we have had 23,707 observations. And when you look at the map, you see, especially when you take into account that we're looking and talking about globe countries, that that is a lot of coverage. But of course, we need more coverage, so we're looking forward to having more observations. And if we can have the next slide, please. Speaking of more observations, in June, people were really out there collecting observations, and we will talk about safety next, because of course this is a time when we all have to make that our top priority. But we were delighted to see that we had 1,192 observations come in June. You can see where most of those observations were from by looking at the map. And we had 61 mosquito larva identifications. We're going to be learning a lot more, doing a deep dive into how to identify larvae today. So we're hoping that many of you will take that challenge on and will give mosquito identification a try. The next slide, please. During these challenging times, let me move this chat out of my way for a second. It is, of course, extremely important that you take your health first. That's top priority. Always follow the guidelines from your local officials. If it is not safe or if you cannot go outside, there are lots of activities that can be done inside. You can access all of the globe data, the data I was just showing you, and lots of other data besides, by going to the globe website. There's the link taking you directly there. It's a really cool way to see all of the data that's coming in from all over the globe. And learn more about Globe's response to COVID-19 by going to the link that's there. You could even put in a Google search and put in the Globe program, or go to the Globe program site and put in coronavirus, and you'll get more information there as well. So we hope you're all being safe and staying positive, and we look forward to, if it is safe, getting your mosquito habitat mapper and land cover and tree and cloud observations. All right, thanks so much.
So hello, everyone. I'm Rusty Lowe. Do you have the slide about Anita on here?
No, I took that out.
Oh, okay. That was what I was waiting for.
Sorry.
So I'm really happy to show you today a live demonstration of identifying mosquitoes. We normally do this live on our own, and our team, which is spatially distributed, Cassie is in South Dakota, Dorian is in Maryland, I'm in Colorado, and none of us have mosquitoes around right now. And there's a lot of reasons why that might be the case. I think where I am, there probably just aren't a lot of human blood meals right now because everybody's inside. But because we were not able to find mosquitoes, I reached out to one of our colleagues, and this is the scientist Anita Schiller. She is the director of the Biological Control Initiative for the Harris County in Texas, and she runs a mosquito nursery for the research that they do. And so what I asked her to do is if she would do a video showing us the different parts of the mosquito, and she couldn't be with us live today, but she sent the video, and then that is really good because we can stop and discuss as we like. And so what I'm going to do now is I'm going to show the video, which is going to show a lot of the different features, and I'll be talking about it. And please put your questions in the chat, and we can maybe deal with some of these in real time. So Cassie and Dorian will be watching the chat, and I'm going to now share my screen. Okay, so does that look good to everyone? Can you see the mosquito larva on your screen?
Yes.
Great.
That's wonderful.
Great. Some of the ones that are, one of the things that was a problem with that we identified during this is that while she has a very clear picture when she's looking under the microscope, she doesn't always have as clear a picture taken with the camera. And so some of these photos are just a little bit blurry, but they are so good. We can still see the measurement, the different characteristics we're looking for. And when we have to look closely at some details, you'll see that we have this a nice view. So let's watch some of these little guys wiggle around. So here we have just a bunch of different mosquito things. And the first question is, which one of these is which of these are mosquitoes and which of these are imposters? So please go ahead and take a look. You can also put some of your ideas in the chat and let us know what you think. So what we have here is she's saying, can you find mosquito larvae? Okay, so take a look at all these here. And what we have here are 3 mosquitoes and 1 larva. So this one that's at the top is one of our mosquito imposters. It is a phantom midge, and so you can see that it's a little bit like the others, but it doesn't have the separate head. So this is one of the features here that we have that is really important. See how the head is round, and it has a neck. Its neck is actually separated from the thorax. And that is one of the features you're going to want to look at, because you have these sympatric organisms that you might find in a mosquito habitat. And so you'll take a look here. All of these are mosquitoes. They look really different. When you're looking for mosquito larvae, you say that this is a mosquito larva. It has a head, it has a thorax, it has an abdomen, and it has a siphon at the end of its tail. This one has a head, it has a thorax, it has an abdomen, abdominal sections, and it also has a siphon at the end of the tail. So it's really cool to see these. She's working with these live, and so it's really kind of fun to see them moving around. I wanted to show you, I'm going to just back up a little bit, or maybe, yeah, let's see if we can get here. So what she's showing us now, and I'm going to stop here, and I'm going to show you, now I'm going to show you in my share screen vision. Okay, let's take a look at this right here. And so what she's pointing out to us is this identification key. I don't think any of you have seen this yet because this is a draft key. We haven't released it. And what we are trying to do is we want to put more pictures in here at each of the steps to help people with identification. But you'll see here in the circle, we see all the different features that we're going to be talking about. So let's take a look at these very carefully. This is the end of the mosquito here. We can call this the mosquito tail end if you want. Usually there is a siphon. If it's not Anopheles mosquitoes, you will have a siphon. We'll be looking for that siphon, that long siphon. There might be some little sort of like it's like a comb, and it's a series of little ridges. And this is called the pecten. That is one of the stages that we need to see when we are identifying mosquitoes. Then we have the terminal abdominal segment right here. There's usually a dark feature right here. We call this the saddle. If you can look at that, you can kind of imagine that that might be a saddle on a horse, sort of sideways. That's why they describe it as a saddle. And what we want to understand is, when we look at these mosquitoes, is does the saddle go all the way around the abdominal segment, or does it go just part way, as we see in this picture? There's also a bunch of hairs that come out. It's called the anal brush. This is a feature that the mosquito uses as it is moving through the water. Sometimes that anal brush is not connected to the saddle. Sometimes it actually pierces the saddle. And that is another feature that we need to look at in order to identify our mosquitoes. So if we look at this identification key, we'll see that we start here, and you find a mosquito larva. You see whether or not there's a siphon, and then we're going to see what shape the siphon is. So I just wanted to refer to this as we move forward, because you'll see that in our video that we're seeing with that she has provided for us. She has given us those options to, she's showing us essentially a great big view of that right here, right? So she's showing us that little piece of that. So we're gonna move on now. Is there a siphon? Good. But I wanna move back because there's something very interesting here that I just wanna show you. And that is this mosquito, just a minute, I'm gonna find it here. Here we go. Just let these guys move around a little bit. We're gonna move over. She's showing us right there, that's the tail. And then I wanna show you this little guy here, because this is pretty interesting. This is a mosquito that you may not often see in your specimens. And one of the things that we talk about when we talk about identifying mosquitoes, different from the imposters, is that most mosquitoes have a round head or maybe an oblong head, but very rarely something that looks like a square head. And this head here is a little bit squarish. So this is a very specific kind of mosquito. This is actually a mosquito called Toxyorhynchides rudilus. And you may know these mosquitoes as cannibal mosquitoes. They are mosquitoes that are friends of ours when it comes to doing mosquito control. And I'm going to show you a really cool picture of this right here. This is one of those Toxyorhynchides rudilus. And you can see they're usually quite a bit bigger than other larvae, and they are predators of larvae. And as a result, one of the things that Anita Schiller is doing in Harris County is she is doing an experimental release program with Toxyorhynchides rudilus as a way to control those mosquitoes that are otherwise problems for people. The Toxyorhynchides mosquito gets all the protein it needs from eating protein when it is a larva. And as a result, when it emerges as an adult, these are also called elephant mosquitoes. When they emerge as an adult, they have no need for a blood meal because they have gotten all the protein they need to make their eggs during the larva stage. And so you might see a Toxyorhynchides rudilus flying around, but they are not going to bite you. So I just wanted to show you this picture because I thought it was a really good one. And it just shows the sort of square-headed mosquito larva attacking another mosquito larva. Okay, so I'm going to go back now to the movie. So what you just saw was this little guy in action, right? And notice the square head. And so very often people might not think this is a mosquito larva because of some of these features that are different, but they are just a very unusual kind. It's only this one genus that looks like this and has this cannibalistic feature. Okay, so there he is. All right, let's see, let's get rid of that. Okay, all right, so she's pointing out here that there is, we know that that's a mosquito because it has an anal segment and it has this wonderful siphon. Here is one here, you can see the head, the round head, the neck area, the big thorax. Here's the long siphon. Anybody have an idea what that long siphon might be in terms of the species? Go ahead and put that in the chat. And you can also see the terminal abdominal segment. So now we're going to take a look here, and she's going to go through the key, and it's like, yes, there, you know, it looks like it has the features. Is there a siphon? So now we're going to take a look at the siphon. So this is going to be kind of fun, I think. So here we're now looking at this one specific specimen. It's got a nice head, thorax, abdominal segments. It's got a clear terminal abdominal segment. We call it the tail of the mosquito, the tail part. And you can see there's a little bit of a, I can point out, she's pointing out as well. But you can see that there is, right there is the we have the segment for the siphon. And now we're going to say, what shape is this siphon? So let's go back and look at this one mosquito that she has selected for us to go through the whole key with. And for some reason right now, I'm not seeing the questions in the chat, but I'm sure that Cassie and Dorian, you can answer the questions as we go forward because they are equally experienced with this. So she's saying, is this actually a cylindrical or a pointed or maybe a spindle-shaped siphon? And this is another step in the key. So if we look at that siphon, some people get confused because we say, well, it does look a little bit pointed, but when we mean pointed in the key, they're actually really sharp and they're maybe a little bit hook-shaped. And we don't have that. We have a sort of rounded edge on the siphon. And so we see that there is, I'm going to stop this for a second. So you can see that the siphon is basically not long and thin, but it's in fact what we call spindle-shaped. Now, I'm a spinner, so I know what spindle-shaped is, but you may not be aware. But basically, when you're spinning, you get kind of a fat area and it goes back down. So it's not exactly, the sides are not parallel. It's not particularly long, and it gets kind of fat towards the bottom. And that's how a spindle, in our weaving mills that we had a long time ago in the 1800s, they look like that. So that's where that word comes from. Okay. So that is then, we'll call this a spindle-shaped siphon here. So if we go back and we look at our identification key here, we're at this stage right here where it says, what is the shape of the siphon? Well, if it's really sharp and pointed, it's probably a mosquito that is what we call a Mansonia mosquito. Those guys have a really sharp, pointy edge because they pierce aquatic vegetation and they actually breathe through the plant. So that's a very specific adaptation. And those mosquitoes don't have any medical issues. And so so they don't have any medical importance. So they're not part of our key. But if we go to what shape is the siphon, if it's cylindrical or spindle shaped, we say yes. Okay, so the next thing is we want to know whether or not there are pectin present. And if you look in this picture right over here, the pectin are little tiny ridges, or maybe looks a little bit like a comb, just a bunch of little protrusions in a row. So we're going to look for that now. So I'm going to push this over, and I'm going to stop my share, and I'm going to go back to looking at our movie. So here we have our specimen. we have identified it as having a cylindrical, spindle-shaped siphon. But what we need to now know is whether or not, now that we know that it is spindle-shaped, we need to know if we can see any pectin. So are there any pectin present on this? And so this is where we're going to get a really, really close-up and beautiful view of using her. All right, so I've stopped this just so you can see it, but let's look at these features that we see at the tail end of our specimen. Do you see this little ridge of little tiny hooks or protrusions here? This is a beautiful example of pectin. And it's a little bit hard to see if you have a 60 power microscope, but it's very clear if you have a 100 power clip-on microscope or better. Another thing that we identify that enables us to identify between Culex and say the 80s specimens is how many hairs are found on this siphon. And taking a look, guys, how many hairs do you see on this siphon? And go ahead and put the answer in the chat. So I'm going to point right here. And one of the characteristics of 80s mosquitoes is that they don't have several hairs. Usually Culex will have several hairs. The 80 specimens usually have one hair or a pair of hairs that are located near the pectin. Over here on this side, you'll see this here is the what we call the saddle, and it is this thickened bit of biological material here that is characteristic of this species, and so we can see that the saddle is not going all the way around the segment, but it's going, it's only partially going around the segment. And that is something that is really good to see. You can't really tell from this picture, and she doesn't have a good picture of it, so I'm showing you here, but you can see that the siphon, it kind of breaks right here. And so you would be able to identify that while you're going on. Okay, let's move on. Take a little bit more of this view. Just absolutely a beautiful picture. Go ahead, Dorian.
We had a couple different answers for the number of hair. Would you tell us from your perspective, what are we looking at there?
Okay, I'm going to go back there. And what we see here is a pair of hairs right here. So it's a little bit hard to see. If you had your own microscope and you were turning it over, you could change the focal plane so that you could actually focus and see that that is a pair of hairs. But you certainly do not see lots and lots of hairs. And these things here, which are pectin, they are not hairs. They are little hooks that come off of the siphon. Okay, does that?
That's fantastic. Thank you so much. I know it makes all of us feel more comfortable when we're like, we think it's this, we think it's that, and it's so nice to go through that and then actually do that, that brainstorming together. That was perfect. Thanks so much.
Thank you so much. Yeah, and I really appreciate. I'm sorry that Anita couldn't herself do this live, but for her to provide us with these things, and because she knows the key that we use really well, she was able to just organize this in a way that we could just talk through it, and everybody will have access to this, so they can use it in your classrooms or with your students or with your to review this on your own. And I know that my clip-on microscope does not come this close. I do not get this good a picture, so this is great, a great resource to have. And you won't see these pictures if you look on Google either. They're Google image. You just don't see these close-up pictures this well. So this is a great resource she's created for us.
Which is why this Meet Up and Do Science is so important. Hey, Rusty, while you're here, we have another question. Miriam would like to know, how do you identify the hairs?
That's a really good question, Miriam. Thank you for asking that question. And the way you do it is they look just like hairs. I'm going to stop right here and push back just a little bit. Yeah, okay. So they look just like hairs. And, one of the things you need to understand is you need to understand what you're looking at. And so if you've never looked at a mosquito before and you see those little pectin, those little protrusions, which is part of the pectin, which is this comb-like rough surface on the thing, you might think those are hairs. And so you just have to learn that those are not hairs, they're actually little scaly protrusions. The hairs really look like hairs. They are, you can, if you have a paintbrush or a toothpick, you can move them around and they move around like hairs. So it's hard to tell if it may be hard to do it if you're not able to interact with it personally, but I think you'll have no trouble when you take your paintbrush or your toothpick and you're moving your specimen around. The hairs will respond the same way as a hair might if it was in the drain of your sink or something. I hope that is a good answer for you. Okay, so let's just move on and see another really cool feature. And she's now queuing us up to look really close in. Oh, she has to focusing back and forth. And she asked me, please, to do a really good job of editing this, and so that no one would see this. But this is how it works. You have to go back and forth, back and forth, back and forth until you actually get the right focus. So let's just have a little bit of patience here with her as she tries to get the next feature on the focal plane. And the next feature, once we know we have a siphon, once we know we have pectin, we're looking for comb scales to see if the comb scales are comb scales that are indicative of the kind of species we're looking at. So this is absolutely a gorgeous, we'll call this our money shot. And it's so gorgeous, I'm actually going to take a picture on my phone, on my computer right now. This is the tail end of the mosquito. If you don't know your mosquitoes super well, it's hard to figure this out, but just think that onto the left, what you have is on the left right there, you're seeing the siphon. On the right, you're seeing the terminal abdominal segment. That dark bit there that you see is the saddle. And then the rest of the body, which is not in the focal plane, is going towards the head as you go to the bottom right of the screen. So I hope I have kind of put that in perspective for you.
There was one other question that perhaps you could answer while we have you here. And that is, the question was, do you know what resolution Anita had for this particular video?
Yeah, I'm pretty sure this is 1000.
Thanks so much. Excellent.
It's 1000. And there are one of the clip-on microscopes that you can get them that go up to 1000, but those are more expensive. They're about $12. And we don't really recommend them for use in classrooms because it is so very difficult in a $12 microscope to get that focal plane. We found that students get very, very frustrated with this, and maybe Anita, I'm sorry, not Anita, Inez can talk about that because she has used both both versions of the microscope, the silver one that has the 60 power and the black one that has 100 to 1000 power. And the black one can provide enough resolution to see these scales, but very often people get frustrated and don't get there. So that's why scientists don't use $12 microscopes. They tend to use, I'm sure she's using here at least a $250 microscope that is attached to her computer. Okay, so see this set of little black features right along the base of the terminal abdominal segment. We can call that the tail segment of the mosquito. And these are, just for a second, I'm going to go back and I'm going to look at, okay, good. So I just want to recap where we are. So we started out with, is it a mosquito larva, right? And we saw 3 cool mosquito larvae and 1 non-larva. So we were able to determine pretty clearly what mosquito larvae look like, including looking at one that is a larva, has the features, but looks very different. That's the Toxioranchites, okay? Then we answered the question, is there a siphon? And we saw that 3 of those mosquitoes had a siphon, and the one that was the imposter did not. It could have been an Anopheles, but because the head is not mosquito-like, we knew that it was probably something else, like a chironomid or a phantom midge or something like that. So then we saw that there was a siphon. We determined that the siphon was not pointed and adapted for piercing plant tissues, but instead it was either cylindrical or spindle-shaped. And in the inset picture here, you see something which is long and thin. This would be what a cylindrical spindle would look like. The one that we looked at was sort of fatter towards the. Okay. All right. So anyway, here these hairs are in a clump. They call it a hair clump, a tuft. Here's some hairs over here. These are the anal brush. But what we are looking at are how many hairs are on the siphon. And this specimen that's in this indent, in this inset, is a culex because it has multiple hairs. And if you look here, right at the base of the siphon, right here, you'll see that there is no, see this tuft right here? It asks if there's a tuft like that at the very bottom of the siphon, and it does not. And so since it does not, we know that this picture in the inset is the culex. But now let's go back to our live specimen, and we said, are there hairs on the siphon? And we saw that there was a single tuft. There was a tuft of 2 hairs. And we saw that right on the siphon, right next to the pecten. So the next question was, does this saddle completely encircle? We saw that no, it only partially encircles. And now we are looking at the comb scales. So I hope you guys are all with me. And so the comb scales can be seen in different ways. Sometimes it's just a big, thick, dark plate that aren't exactly separate scales. That's not what we have. But we do have comb scales. So in our key, that takes us to 80s, right here. And so now what I'm going to do is we're going to now determine in our 80s if we have just an 80s we can't identify, or if there's 80s aegypti, or if it's 80s albopictus. Okay, so now we're going to go back to our live presentation. Okay, so I know this is complicated and it's really fun to do this step by step. So as you have questions, please go ahead and ask them. So we're going to go back here and.
Rusty, we do have a few questions here. One is, what is the function of the following features? And the first feature is the siphon. What is the function of the siphon?
Oh, thank you so much for that great question. And the siphon is how those mosquitoes breathe. So if you remember that picture that I showed you right here, and I'm going to stop my share and I'm going to go back into sharing my screen and I'm going to show you this picture. Do you see that right now? You can you see that?
Yeah, perfect, perfect.
Right. So you see that these mosquitoes here are hanging by their siphon at the top of the water. And this is where mosquitoes usually hang out. They hang out there because they're breathing through their siphon and they are eating tiny plankton and things like that are in the water. So they're eating and breathing at the same time. So that's what the siphon's doing. And so that's a really great feature to become aware of. And it also, they tend to be very distinctive from species to species. Okay, another question, Dorian?
Yep, so the next one, then, that great answer. The next one was, can you tell us the function of, and I guess I'll go ahead and just say the next 3, the saddle, the pecten, and the hairs. Okay,
great. And there is essentially, I would say, no significant, these morphological characteristics are not necessarily functional. The same way that, I have gray hair and Cassie has white hair and Dorian has brown hair. It's just the morphological variation that we see between distinctive kinds of mosquitoes. They have evolved in this way over time, and they don't necessarily have a specific function that we are necessarily aware of. They are just morphological characteristics that we can use to identify them. You know, the same way brown hair, blue eyes, you know, brown hair, you know, those kinds of things. They, you know, they don't help us to live in any particular way, but they do have, you know, they do have these features. And this is probably, if you want to go into why, it's probably because of genetic drift, that over time, a mosquito species, which is found in isolation, tends to change over time and create variations, you know, at every generation. And this is the result of it. I hope that's a good answer for you. But you know, the pecten doesn't necessarily have any particular feature, specific trait. The brush, the anal brush, which we find on the saddle, some of them are a little bit bigger and they're a little bit better swimmers. Others have fewer hairs and they are not such great swimmers. But those are just variations and don't really have anything to do with they're not characteristics that ensure their survival.
Fascinating. Thank you so much. And great questions. Thanks.
Great. Thank you so much. Okay, so what we're looking at now are these features here. These are the comb scales. And comb scales have basically two variants that we need to be aware of in order to determine what kind of mosquito this is. So what I'm going to do now is I'm going to show you once again the key that we have, and or maybe I just, well, yeah, I'm going to show. And you'll see that when we get to knowing that it's Aedes, one of the things we need to do is there a tuft between the pecten? So if we go back to the image that we have, and I'll show it to you as soon as I go back to the other image, there is no tuft of hairs inserted between the pecten teeth. And so that's why we know that it's not one of the other Aedes species. It's going to be one of the two baddies for human health, Aedes aegypti or Aedes albopictus. So now what we need to do is we need to look at, and we're going to have a discussion about that. And I see that two people are raised their hands. Go ahead and put your comments in the chat and we'll talk about it. And do you see my mosquito? Do we see the video now?
Yep, looking good.
Okay.
Really nice image of those comb scales.
Yeah, so does anybody have any questions before we move on?
I guess the question was, what would the magnification need to be in order to see the comb scales?
100 power.
Cool, thank you.
Yeah, okay. So this has taken quite a bit longer than I actually thought, and I really apologize because we had some other great things planned for you today. But I think this has actually been very useful. We've had some great questions. But the answer is, do you think this comb scale, if you look at it, is it long and thin like a point, or do you sort of see that it's kind of triangular and then you have a point coming up? And what we're actually seeing here is something which looks a little bit like a pitchfork, like three different pieces with a long, thin middle attribute, and then two shorter attributes sticking out. So based on these comb scales, we're going to call this an Aedes aegypti. And if you look at the pecten here, you'll see that there's no hairs that are inserted between that pecten. So I think we're doing pretty good here. All right, so we can now answer some more questions. And then I would like to go on because we have created for you a really cool quiz.
Rusty, some people could not see the pitchfork. Could you, can you show us that again? Yeah, this is so helpful.
Yeah. I want to show you this one thing here too, because it's really, this is one thing that people don't realize, is this vision here. See, this is the one tuft of the Aedes aegypti, and you see it has the pecten. And the pecten, actually, there's two rows of pecten. When there's one row, there's always two rows. And it's just really hard to get that view. And she tried really hard to show us that there are those two rows. But let's go back to those comb scales, and I will show you. It's not particularly, I think that what we would need to do, and I'm sorry I don't have this queued up, but what we would need to do is we would need to show you, I I think that it'll be very clear to you that these are the pitchfork scales, because like I said, if you look at it, it's kind of like an upside down triangle, a very acute triangle with something sticking out of the top. And if we had maybe even more power, you would see that there's two very little points that come off of each of those sides of that triangle, and then it has a long one and thin. When you have a Aedes albopictus, you just have one long, thin comb scale. So that's the best I can do to describe that. But this video is quite a bit longer, and it has a lot of other features that we can take a look at. So she did a really good job of going in and looking at these different features and showing us what we need to see in order to do those, in order to do our identifications. So there we go into focus. There. Perfect. There we go. And so here is just a last view of this one particular specimen. You can see the comb scales down here. I wanted to show you. I thought that you might see them a little bit better, but you can see here that it's just not a long and thin scale. It does have this sort of what they call a pitchfork shape. Once again, less clearly because we are focusing in on basically right here, but you can see that there are the pecten, the hair right here. And so I think we're good. So I'd like to take some questions, but I thought maybe what we can do is, Cassie, with our C students today, we had them put up their mosquitoes, and we thought we would end up with a mosquito quiz that has images of mosquitoes that have been found by our high school interns this summer. And I want to answer, now I can see the chat when I'm not presenting. Why does the color of the mosquito larvae vary? It's because of the transparency. It's because of the kind of water that they have. But usually where you have thicker, a thicker material, like on the head or on the tail, you tend to have it darker. So anyway, this is great.
In general, the questions, even though in the poll it's worded, is this a mosquito larvae or an imposter? So is it a mosquito larvae? It's either no or yes. In the first picture right here, submitted by Frank, mosquito larvae or imposter. And here's the identification key Rusty was using. So here's number 2. Is it a larvae or an imposter? So yes or no to a larvae. I'm going to say this is an imposter.
Mm-hmm. I don't know if you ever did this, but I remember this little guy from when I was like in 6th or 7th grade. It's a planaria, right? Because yep. you can cut them in half, and they're bilaterally symmetrical, and they will grow the other side. So I remember that. So okay. Thanks to Bzzz for that one.
Yep. And the third one, a mosquito larvae found in this pond with a lot of detritus on the top, submitted by Rael. Is it a larvae? Yes or no?
Great. And one of the questions that Grace had was, is the tail the only thing for identification? And you can actually use a lot of other different features, but we wanted to make it easy for citizen scientists. And it turns out the medically important species can be determined just by the tail. You can also use other features if you use another key, the head shape, the kind of hooks. There are other features that can be used to accomplish the same objective. But in our app, we use the tail segments for our identification.
We're all experiencing all kinds of amazing and frustrating things with our doing everything virtually, and it could be that
I think she's dropped off.
Well, we'll figure out how to carry on. I will say, when you look at the questioning, we had so many great questions. One of the things that it might have been a little challenging to try to answer, right? Was it a larva or not? Those were great images. Way to go, C students for grabbing those. Now it looks like Cassie is back, and so we'll see the result of our polls here. Mm-hmm. There she is. Awesome.
Awesome. Thank you, Cassie. It's nice to not be the only one that has technical difficulties today.
Every day, all the time, everywhere. We're all creating this plane as we fly it.
One of the great questions here that Kushi had, Patel, was the environment of the breeding site affect the type and variation of the morphological features? So in the long sort of evolutionary adaptation world, yes, they definitely do. For instance, maybe the color, maybe the mosquitoes are found in a place where the water tends to be murky and therefore they have slightly darker color, because they are the darker ones are the ones that are not immediately picked out by frogs or birds. So yes, absolutely. Another question that came up here about the variations, maybe from precipitation patterns or temperature variations. And, I can't answer that because we weren't there over the thousands of years that these mosquitoes were going through generations after generations and changing form. But I will tell you that the features that they have do relate to the kind of habitat that they live in. And so there will, the Aedes aegypti and the Aedes albopictus mosquitoes are both container mosquitoes. And so they have attributes that are that help them to survive in that kind of environment. Here's a great one. Is this a mosquito or is this an imposter?
And we don't have the poll up right now, but feel free to just type your guess in the chat, and Rusty will reveal the answer in just a moment.
Mm-hmm. So I'll answer you here. Look at this. It's kind of like a squarish head. It's got a funny little mouth here. But this is not. This is supposed to be. It's supposed to be one of our imposters. It is. I picked this one out of the. Out of this the selections from the students, and this one is one of the really hardest ones. I would actually say nope, it's not a mosquito, but then I would look at the it does have a siphon, it does have an anal segment, and so this is actually a Toxioranchides rudilus, which are the weird ones that don't look like mosquitoes. All right, this is not a larvae, but one of our students found an Aedes albopictus mosquito, so I thought Aegypti mosquito, so I thought I would share what it looks like. Okay, I think we're almost done here. Okay, this is a really good one. Is this a mosquito larva or an imposter? So think about the features. Does it have a round head? Does it have a separate abdominal section, which is sort of broader than the head? And do you see a siphon? And the answer is no, that this is one of those imposters. This is one of those midgy fly. Here's one. This was one that a lot of our interns argued about. Is this a mosquito pupa or something else? And the answer is yes, that was an imposter, but this is not. This is actually a mosquito pupa. You really can't tell much about it because it's undergoing metamorphosis. This is what happens inside a chrysalis for a butterfly, but these happen, right in the water. So this is a mosquito pupa. But if you look at the next slide, someone said, "Is this a pupa?" And what do you think? Yeah. So this is actually an ant. This is an ant up close. So this was a pretty cool one too. So you can see that, when you're looking under a microscope, especially if you have not the whole body under the thing, it's hard to tell. And here also is another one of these imposters that has the head that's bigger than the abdomen, and it does not have a siphon, but otherwise has a very sort of mosquito larvae-like body. So that was another imposter. I think this is our last one. What do you think of this one? Can you tell me what kind of mosquito this is by looking at okay, I just told you it's a larvae, sorry. But can you tell me what kind you think it might be? Do you think this is an Aedes, or do you think this is a Culex, and why? And we don't, we'd have to be sure, we'd have to go in and look a little bit more detail and look at the hairs on the siphon. But what do you think? Yes. So this is a Culex because it is long and thin. The sides are parallel on the siphon. And if we were able to see that, yes, there are a few hairs, and no, there is not a tuft of hairs at the end of the siphon, we would be able to tell for sure. Excellent job. Okay. And this is another thing that someone submitted, and you can see it's got legs. So that's not going to be a larva either. So great. We do have 3 or 4 more minutes, and I wanted to give Dorian an opportunity just to talk about how and why identification of mosquitoes is important, how it can play a role in in doing a scientific project. And here she's going to present one of the really cool IVSS projects that that were submitted this year.
Thank you so much. Yeah, I do hope that a lot of you have seen this, and those of you who do work with students have said, wow, I'm going to take it to the next step. Definitely worth having some of those magnifiers. The students are going to love this. If we go to the next slide, please. This is one project in particular, which was looking specifically at the larva. You can see here where this was submitted from. I just have to move things around on my screen. So you can see the location of the school. And if we have our next slide, please. This was the poster that the students submitted. You can see their nice images there of their larva. We won't try to look through this and find all the results, but if we'll go to the next slide, I believe, never mind. In this slide here, basically what you're seeing, and you certainly, anyone has access to all of the student reports. You go to the GLOBE program website, go to community, and then go down to annual symposium or the International Virtual Science Symposium and look at the student reports. They are wonderful. We will focus on student reports in another webinar later, but we did have several reports that, like this one, focused in on the different larval species that they were finding, and then developed recommendations based on what they were finding, which type of genera, species that they were finding. They came up with some recommendations for their school as to how to reduce the number of mosquito larvae there. So really, really good work, and congratulations to this group in particular for being featured with the GLOW program. And last slide.
We invite you to our next webinar, August 13th. The registration link is right there, bit.ly/Cs-IVSS. So we'll be sharing some of our Cs projects along with other submissions from the International Virtual Science Symposium, giving you some ideas that you can plan your student investigations throughout the next school year. And I think they open up in January, don't they, Dorian?
That is correct. Yep. But you know, it's always a good idea to get going on them as soon as possible, because you know it's just great to be doing your research. For many of us, at least for myself in Damascus, Maryland, we don't have active mosquito season all year long. So it's nice to be collecting data now, even though we don't submit our reports until, like Cassie was saying, you can start some in January and you can continue through usually the beginning of March. It's a great idea to be thinking about what kind of project you'd like to do, considering the different GLOW protocols that you might use to do this project, and starting to collect your data. So thank you so much.
Thank you very much, Dorian, Liz, and Rusty. The archive of the webinar will be up on our Globe Mission Mosquito campaign webpage in about 48 hours at the very latest. And the slides will be up there also.
Yeah. I just wanted to say, too, that about the IVSS projects, the high school interns, the NASA high school interns that we're working with this summer are working all summer. They have started in June. They'll be ending at the end of July, but they are preparing, all their projects will be submitted to the IVSS in January, because you know they won't be able to do mosquitoes in the summer, in the late summer, early fall. You know, that's when the mosquito season will end for them here in the in North America. And so you know, you can do these projects anytime, and they're just going to hold on to them. You know, maybe they can do a little bit more, you know, background research or something, but their projects will be done by July. And so we will be presenting some of those in the next webinar. So I really hope you can make it and get some great ideas for what you might do with your students.